Okay, so welcome everyone. This is the last two lectures of the course. And so we're finishing off the major groups of plant pathogens. So today we're going to talk about nematodes. Interesting little beasts they are, and fairly neglected when it comes to really their impact on plant productivity and their impact on plant disease. Nematodes are often the hidden disease. You know, sometimes they're fairly obvious, and then they're a clear disease. You know, occasionally the nematode lesions, particularly the cysts or the melanogoidy nematodes that actually cause lumps and bumps on some of the harvested produce. They're fairly obvious. But a lot of, particularly the lesion nematodes and those like that nibble away at roots and don't kill plants outright, can cause quite major yield losses without anyone really noticing it, because they tend to just stunt plants a little bit. And it's only when people with an interest in nematodes and nematology have actually demonstrated eliminating them that the influence on yield and growth becomes more apparent. So these guys are an underexplored and underestimated impact on agricultural production. We don't have any nematodes in Tasmania because we don't have a nematologist, so no one actually bothers to look at them, but no, there are plenty of issues. Anyway, nematodes. So what are they? They're essentially unsegmented worms, so they're a worm-like structure. They're an animal. They're not a fungus, they're not a bacteria, they're not a virus. They are an animal. They belong to the phylum Nematoda. Sometimes they're called roundworms, sometimes they're called eelworms. So obviously when you drench your farm animals or your cat or your dog for roundworms or those sorts of things, you're actually trying to kill nematodes, parasitic nematodes of animals. So there obviously can be parasitic across a wide range of things. The vast majority of nematodes are not parasitic though, they're actually free living, and particularly in the oceans there's actually a hell of a lot of them, but also in the soil and in other fresh water. They are aquatic animals, they need a moist environment to move and survive in. Most are pretty small. And that's kind of why they fall into the gambit of plant pathology, because they tend to be microscopic in their mode, as opposed to something a bit bigger that probably falls into entomology when it's an insect or insect-like organism. They are very abundant. Some of the stats here. If all the nematodes in the Murray River were combined into one animal, it would be over 20 metres long, so that's a bit scary. I think there was a comment that if you removed all of the matter from the earth, and except for the nematodes, you actually still would see the earth, because there'd be a complete skin of the planet made up of a layer of nematodes that fully covers the whole planet. There's a lot of them. What do they do? This is roughly the breakdown. As I said, most of them are marine. So about half of the nematodes, at least they know about, live in the marine environment. There's a big bunch that are involved in eating microbes and part of that food chain. So you've got plants and other organic matter that gets degraded by fungi and nematodes, and perhaps directly by fungi and bacteria. Sometimes directly by nematodes, but the nematodes are also important at cycling the fungal and particularly the bacterial in the soil, eating those guys up and converting those into other food sources for higher organisms. So nematodes that get predated by arthropods and the likes, and then into the higher organisms. In terms of agriculture, well, not all agriculture, but about 25% of them are parasitic. and kind of half and half between animals and plants. So we're going to be interested in the 10% that are plant parasitic, and that's the ones we're going to mostly talk about. Obviously, nematodes have a major role in that food cycle, in decomposition of matter, release of nutrients, and again, some of the stats there. So about 30 to 50% of nitrogen that crop plants release are made available by bacteria and by bacteria-feeding nematodes. So they actually are responsible for cycling a lot of the nutrients in the system. This comment here, this is interesting because this is becoming more and more important in our trying to understand and analyse soil and soil health. The whole concept of soil health is very trendy. Everyone wants to have healthy soils, but no one can really define them terribly well. What is a healthy soil? It's a productive soil, but how do you actually measure that? And there's a number of potential measures you can use. One that's actually quite useful is the abundance of free-living, so non-parasitic nematodes. So how many of these bacterial-feeding nematodes are in the soil? Why is that a good measure? Because these guys are fairly labile. They don't tend to, they tend to die fairly easily. Bacteria can be pretty tough. Fungi can be very tough. Other microbes, if you look at soil health in terms of biology, can be toughish. You can dry them out and they're still there. Nematodes die fairly easily. So if you've got a hard-worked, fairly non-productive soil, it's likely to have much lower free-living nematode content than a really vibrant living soil that's got plenty of microbial activity. It'll have plenty of free-living nematodes. So there actually are now people working on specific diagnostic tests to measure the amount of free-living nematodes as a surrogate measure of soil health. Anyway, that's an aside. Some are obviously parasitic. Some of them actually are parasitic against insects, entomopathogens. We used to have in fact, out at Newtown where I work, there's a building there. It's still called the insect path lab, where they actually set up a program for breeding up nematodes for killing insects in soil, particularly corby grubs and cockchafers. The guy who set that up disappeared off and set up a move to the States into a biocontrol company there, and he's subsequently come back and selling chemicals, I think, of all things. But anyway, so a biocontrol program was fairly active in Tasmania at the time, breeding up these little beasts that happen to be quite good at killing some insects that might be hard to kill in another manner. And here's some examples of where they've used nematodes in a biocontrol program to control certain pests. So Cyro, that's not necessarily Tasmanian an example, but for control on pine plantations. They also have been useful for research in animal systems because they're small and easy to grow and people don't care too much if you squish them, as opposed to big, nice furry things. And it was the first animal to have its full genome sequence. Nowadays, genome sequences are less exciting because everyone's doing them, but it was certainly the first animal to have a genome sequence. Right, the parasitic ones. So there are a number of parasitic ones. Those that attack humans and livestock we kind of tend to call roundworms, but they can be hookworms, roundworms, pinworms, etc., including some very nasty ones that cause some pretty nasty infections. So some of those more tropical ones that Google them on the internet and put you off your lunch as to what they kind of do when they crawl inside your leg. Some of those movies you see where things are just not go there. Various livestock parasites can suck blood, cause anemia, can be fatal. Others can cause scars. So this is why drenching is an important part of livestock management in animal production to control your nematode parasites. But this is what we're interested in, the plant parasitic ones. That's what we're here for. This estimate is a very crude estimate, and I would guess grossly underestimates the actual physical losses. Now again, this loss is in productivity, not losses in terms of all of those other aspects of loss we discussed in the first lecture, which would increase the dollar value of that and the impact of that to much higher levels. Most of the good estimates of this come, in the Australian context, come from cereal production, where they've got a reasonable handle on the role of nematodes, because they are a pretty major issue in cereal production. But as I said, I suspect that the impact of nematodes globally, and particularly in Australia, are grossly underestimated. They cause damage by direct feeding. They can also interact with other pathogens. They can increase the susceptibility of the plant to invasion with other pathogens, or in some of them at least, there actually are the specific vectors of plant viruses. So we're linking our virology and nematology here. So a bit like with our viruses, we're mostly talking about our flying insects, bringing them around. There are soil-transmitted viruses that are transmitted specifically by certain nematode species, and that becomes quite problematic. You get the nematode effect as a parasite, and you double that with the virus effect. Gross morphology, what do they look like for the parasitic ones? I mean, a really important part is the mouth part in this stylet. That tends to highlight that you're dealing with a plant parasitic nematode. All the bacterial eating, consuming nematodes have a completely different mouth part. They obviously just open up their mouth and suck in bacteria. The plant ones need to penetrate through plant cell walls, so they have this stylet structure, much like some of those aphids and the likes have, something that's sharp and can penetrate through cell walls. So they penetrate through to actually enable feeding in a plant system. They don't have a blood system, but they are fluid filled, and that's how nutrients move around. They're essentially one big digestive system in a way. They suck in stuff, they've got a pump here to move it through, one big intestine, an ovary to produce more of themselves, and then pump out the stuff at the end there. So just an efficient food factory. They don't have lungs, they don't use respiratory organs, but they use diffusion to breathe. So it absorbs oxygen by diffusion through their system. And they don't have a blood system, but the nutrients move within the fluid inside their system. How do they grow? The outer cuticle is fairly tough and fairly strong. So once they outgrow it, it's a bit like a snake. They kind of shed their skin and they molt and they produce a new version. So like many insects and other things, they will go through a series of molts as they go through different stages from early juvenile stage to second and third stage before they hit to adult stage. How do they move? A bit like other worms, they kind of slide along. They a tube within a tube, so the outer skin cuticle has muscles to allow it to move and then pulls the inside along and inches its way along. Obviously they don't have legs or flagella or the likes. And how far do they move? Well, that there's an estimate that they can go maybe in their lifetime up to a metre. So that's a reasonable distance for a little fella. And that highlights why when you're actually looking at nematode diseases in soil systems, you'll often see it as patches. You'll often, if there's an initial infestation, it'll slowly spread, and you'll get a patch of stunted plants as that spreads, because they've got the capacity to move to adjacent plants, maybe a second one along, but they're not likely to go to the far end of the field like a windblown spore. So, spread and movement is slow, pending the movement of that actual soil system. So if you're actually transporting infested soil, obviously that's another way of moving them around a bit quicker. How do they survive? They try and avoid being eaten. So either stay still, some of them produce cysts, so robust structures that will actually remain viable even under desiccated soil and other structures. Some of them also survive inside their host plant and actually can overwinter in that manner. You can, in terms of taxonomy, and I'm not particularly going to go into any detail with taxonomy of nematodes because it's a discipline all by itself, but a big part of it is this internal structure. So the shape of the mouth part and the esophageal structures can split them into the different types of nematode. Why does that become important? Well, obviously the different nematodes have different feeding structures, and particularly if you're dealing with certain ones that might be vectors, you need to know which species you're dealing with because they have some quite specific associations with their virus vectors, with their viruses. There's a whole bunch of them, and they vary in size from bigger ones like the Longodorus through to the Pratelinchus, the lesion nematodes, that are fairly tiny. Most of them are worm-like. These guys here, the cyst nematodes and the why has my mind gone blank? What are they called? Oh. I'll remember in a minute when I stop thinking about it. Root knot nematodes, that's what they are. Yeah. So the cyst nematodes and the root knot nematodes, the females are sedentary and form this big round pear-shaped type structure. The males will still have a worm-like structure. So you do get some differences in their structures and morphologies. Most of the plant parasitic ones fall into the Cycloida order, but there are some in others as well. So here's a list of some of the important plant parasitic ones. As I said, the root knot ones. I should have gone to the slide and I would have remembered. Melanogoi root knots, the Globigerina and Heterodera, cyst nematodes, Pratelinchus, the lesion nematodes. They're all really common and quite nasty. Dodolincus, stem and bulb nematodes. That's also quite important here in Tasmania for onion and bulb production. You've got dagger nematodes that are also these guys are the astraces. They're important parasitic ones. They're also the ones that are virus vectors, and a bunch of others. I won't go through any more detail. Some of the important ones, the more common ones, the root knot nematodes. So these actually cause visible lesions, visible structures on the roots, visible galls or on the roots, and a whole range of different crop species can be affected by these. Cyst nematodes, these form, rather than a feeding structure on the root, they're actually a cyst, a different type of structure, but similar in a way, on the root system as well. The lesion nematodes, these guys just punch holes in roots and actually cause small and invade inside internally in the roots and actually damage the root function in that manner. And again, tend to be the Pratelinchus species. And there's a huge, that's just a couple of examples, there's a huge range of pathogens, of crops that are affected by lesion nematodes. Crudely, its life cycle goes from adult, producing eggs, goes through a number of juvenile stages with molts in between, feeding, causing damage, surviving. So surviving, and the survival mechanism tends to be either as a robust structure, a cyst, or an adult that can survive in the soil for a short or a long period of time, or alternatively within the plant systems themselves, in plant roots, where they can survive in alternate hosts for a long period of time. So what do we happen? So first stage, molts. Second stage starts to hatch. The egg hatches. They start to feed, and then molt again. Feed and molt, feed and molt, feed and molt, and eventually lay eggs. So, kind of, what does a nematode do? It eats and makes more nematodes, and grows a bit bigger each time. How does it feed? As I said, this is the important part, the stylet. So the stylet, or sometimes called the spear, is the diagnostic factor. If you're doing a nematode extraction from a soil sample and you want to know if you've got parasitic nematodes, you're looking down the microscope to see if you see anything that's got something like that, and then you know, okay, I'm dealing with a plant parasitic nematode because it's got a stylet, and that means it's designed for punching holes in plant roots and eating plant material as opposed to all the others. There are a range, we can define the plant parasitic nematodes into major groups based on how they feed. So there's those that are the ectoparasites. They live outside the plant and feed, so they penetrate from outside and they feed on the plant root systems with only their stylet or maybe a small part of their head inserted into the root, but mostly external to the plant. There's the semi-endoparasites. They usually have a bit more of themselves, not necessarily fully embedded, but most of themselves embedded into the plant tissue as they're feeding, so they can be stuck there. And then there's the full endoparasites that actually go internal. They penetrate inside the root and live fully within the body of the host they feed. Whoops. Within these, some of these are what we call sedentary, so they don't move. So they get in, particularly some of the endoparasites, they get inside, they remain there and they feed there for their life. They don't actually need to move and they don't bother. Others are more migratory and move from site to site, and particularly a lot of the ectoparasites that will actually feed and then may move and feed again on a different part of the plant or a different plant altogether. Some pictures. So here's some external feeding ones, so images at least of your nematodes actually penetrating the roots, these ectoparasites, and gaining nutrition from external sources. Again, another microscope image, so inserting the stylet and feeding from the root here. Versus the endoparasites. So here's some lesion nematodes that are actually inside the root. They've been stained so you can see them, but you can see there's quite a number of nematodes within that root, obviously impairing root function quite dramatically. So the nematodes feed on plants, derive nutrition from plants, and that's a major part of how they cause damage to plants. But I also mentioned how they can also interact with other pathogens, and that can be equally important in terms of their impact. So they may make a plant predisposed to infection by another pathogen. It might be that the actual damage of the nematode, the feeding damage, provides an entry point and allows penetration of that plant with another pathogen. That might also be that they can interfere with the host defences, and in doing that actually make the plant more susceptible to another pathogen. So here's an example. Here's one of the root knot nematodes in tomato, and it can exacerbate infection by Fusarium, so a soil-borne fungus, Fusarium oxysporum, by I think in this instance by creating entry sites and allowing infection. You can get occasionally the opposite, where disease severity might be reduced if there's an antagonistic interaction between the nematode and the and other pests. Less common. Where that, in this instance, we're talking about where the nematode will actually directly interfere with the other pathogen. In this instance, this is more indirect interference, but actually can also exacerbate, can also decrease disease, where you've got your nematode. As it interacts with the plant responds to that infection by switching on defense processes and saying, okay, I'm being attacked by something. I'm going to switch on the jasmonic acid defense pathways, and I'm going to slow and try and stop that damage by this entity, which is a nematode. In doing so, that same defense processes will also potentially inhibit infection by maybe a soil-borne fungus. So they might provide entry points and exacerbate disease. Sometimes they'll switch on, they'll pre-induce the defense responses of a plant and actually make it more resistant to a fungus. So you really can't determine what that interaction is going to be until you test it. And we already mentioned some of them can be virus vectors. So the Ziphinema longidoris, trichodoris are the species that can be virus vectors. And there's two major groups of viruses: the nepos, spread by Ziphinema, and tobras by the trichodoris. And the longidoris. In terms of those as virus vectors, the trichodore, that's the rod shape, that's the tobras, and these guys, the isometric nepoviruses. These viruses, they're all really broad host range. They all invade through root systems. And so they are well adapted to associate with a nematode that can feed on a wide range of host species and infect a whole range of host species. So why these become important? These are actually, there's a lot of, particularly the nepoviruses, but even the topraviruses, a lot of those are of tremendous quarantine significance because they cause pretty nasty diseases in a wide range of hosts. A lot of them are not present in a place like Australia because they tend to move with the vectors. And we talked before about how nematodes are relatively labile. So if you're trucking produce back in the 1800s from the UK to Australia, and there might be soil attached, bringing fungal pathogens occurred fairly frequently. So a lot of the pathogens we brought over with the early transport of all our crops came in the ships that sailed for months with carrots or potatoes or whatever else in them to actually grow our next set of crops. And there's bits of soil attached and all those fungi and whatever else carried over with it. A lot of the nematodes didn't make it because they are relatively labile and they didn't survive the months of sea journey. So we're fortunate in Australia that a number of these nematode pathogens and indeed some of these nematode vectors are missing. So as such, some of the viruses too. But even when later on we've introduced the virus by bringing in infected plant material, it's not spreading because we don't have the nematode present. So yes, anyway, so these guys become increasingly important in terms of quarantine because some nasty diseases and currently not present. If we look at the virus vector association, just because we kind of looked at this briefly with our aphids, these guys are a little bit similar to the non-persistent transmission with aphids or semi-persistent transmission, where they bind to this mouth part. So either in the stylet region or just below the stylet region where the virus is bound. So when the nematode feeds, the virus sticks to that mouth part, and then when it feeds again, it spits it back out again and transmits it in that manner. The association can be relatively short or can be quite long depending on the longevity of the vector itself. But will be lost during molts. So in that life cycle, depending on when it's acquired, if it's acquired as an adult and then goes into a cyst mode, it'll survive for a long period of time. it's acquired as a juvenile, you'll lose it every time you go through a molting stage. Right, anyway, diagnosis. Nematodes are often ignored, and I'll keep saying this because they tend to be ignored. A lot of the damage they cause is not obvious. If you've got a fungal leaf spot, that kind of sticks in your face and goes, oh, I've got a leaf that's got a spot on it, I don't like that, let's spray it, make it go away. If you've got a few nibbles at a root and the plant is decreased by production by 10-15%, you may not even notice it, but that 10-15% might be really, really significant in terms of yield. And that tends to be what nematodes do. They don't want to kill the plant, they want to feed off it in a parasitic manner. They're not really eating dead material, they're sucking from the living plant material, so they don't want to kill plants necessarily. There's some of the other issues with the nematodes. The symptoms, when they do show obvious symptoms, can look very much like other diseases or other problems. So it's root damage, so some of the fungal diseases, some of the bacterial diseases that damage roots, even obviously some nutritional factors and soil structure factors can give similar symptoms to what a nematode causes. This is really important, and why nematodes become important. The management of them is actually really, really hard. Management of them is really hard, but management of other pests and diseases may mask the symptoms too. So by the time you're actually starting to manage some of the other pests and diseases, you actually may also be impacting on the nematode numbers too. But I'll go back to that stage, because that's an important point. Management of nematodes is really difficult, and often when it's been pointed out the impact a nematode's having on a particular crop, that's generally been ignored in the past, and then go, oh wow, that's important, what do we do about it? Tend to put our hands up in the air and go, it's not really a lot. There are some really, really nasty chemicals you can apply, and that's, or you can try and look for host resistance, but there's not a lot of tools in the toolbox for controlling nematodes that are actually terribly friendly. And a lot of the nematicides that are available are getting thrown off the shelf because they're all S7s, they're all incredibly toxic. They're killing animals, so they tend to kill us too. And as such, they tend to be materials that we don't tend to like to use, or the APVMA is looking at quite closely and saying, I don't think we should allow the use of this, it's too dangerous. Anyway, that's an aside. And because the symptoms tend to be diffuse, stunted plants, they're often not. That's characteristic. Gee, that's definitely a nematode. You have to do a lot more work to find them. They're hard things to find. So here's an example. Stunted plant, healthy plant. It could have been stunted by a whole range of things. In this instance, it was a nematode. So how do you discover if you have got a nematode? What's the way to do it? Well, obviously the root feeding, so you're examining roots and the soil around roots. You want living plants, dead plants won't be any use because any dead plants the nematodes will have left. They need to be feeding off living plants. So you want to sample from, if you've got an expanding patch, near the edge of that expanding patch where they're more likely to be actively feeding. You want to sample from the root zone, you want soil from the root zone as well as examining the roots. So you can examine the roots directly, particularly for those endoparasitic nematodes. So you can, as that image showed before, you can stain them and actually look for presence of those. But those ecto-feeding ones, you won't necessarily see that well. Then you're going to be actually examining and sampling the soil itself. So soil and the roots. Above ground plants can be useful. There's some nematodes that actually do migrate further up the plant. So in your soil sampling, again, try and get a suitable soil sample from about 5 to 20 centimetres depth in that early root growth area where the nematodes are most likely to be feeding. So when there's young rapidly growing roots, that's where you're going to have more nematodes actually feeding. And I'll leave you to go through that. That really talks about some of the sampling strategies in terms of numbers of samples versus bulk of samples. More samples that you combine are better than one big sample. You've got a statistically better chance of detecting them. In the end, it comes down to what's practical. You can't, if you're testing 30 or 40 fields, you don't necessarily want to do 300 or 400 samples per field, for example. That's what's practical as well as what's going to be useful. Some examples. So here's, again, you see these sort of patches. There's this expanding patches that are probably more visible from aerial shots. Here again, you can see a patch of plants that are doing poorly. I mean, there's also something you might have expected if it was a low-lying area in waterlog, but in this instance, it's certainly nematodes where they have occurred and they're slowly expanding in that field and causing damage. So how do we find them? How do we extract them from the soil? There's a number of methods. The Bayerman funnel is a common one, and we'll have an image of how that works in a minute. There's other methods where you can actually allow them to settle through. This whitehead tray or pie pan method. You can try and flotate them out and spin them out using centrifugal processes to try and concentrate your nematodes. Where those nematodes form cysts, so these are several millimeters across, you can actually float and sieve those out of soil samples. That's actually easier to detect than the free-living nematodes. Some of them, obviously, you can get directly from the plant tissues and then do direct staining. So here's some examples. The Bayerman funnel. So you've got your soil. You have essentially a funnel with a clamp onto that stage. You add water to it. You pour the water soil mixture in there with a crude filter, and the nematodes tend to fall the way through the filter, leave the soil behind, and kind of accumulate down the bottom. You put a light at the top, and they kind of run away from the light, and then you can release that, collect that into a jar, and then observe that under the microscope. So you've got your soil sample. You can concentrate the nematode sample in that from that soil after a couple of days, allowing them to settle down, and then observe that to see what you've got. That's quite a common method. A sieving method, again, going through different sieves, going through coarse sieves to get rid of your soil material, and then fine sieves eventually to actually capture the nematodes. And then either stick them in a Bayerman funnel and collect them as above. So just to try and get rid of a lot of the excess soil. Centrifugation processes, either in water or sugar solutions, so you can actually float them up and get them floating in the solution, and spin out all the heavy rubbish, which is the soil sample, and then take your samples from that manner. So spin out the heavy stuff, keep those in solution, put those through a sieve, and away you go. Essentially, it's all ways of separating them from the soil and trying to concentrate the number so you can observe them. What do we do about them? How do we manage them? As I said, a lot of them haven't made it. A place like Australia, nematodes, quarantine becomes really important because of the fact that a lot of our pests and pathogens arrived in the good old days before we even had quarantine in place, where ships loaded with produce arrived. But being relatively labile, a lot of nematodes never made it. So we are thankfully free of a lot of nematode pathogens, and we want to keep it that way. So how do you keep it that way? Obviously nematodes move particularly in associated with soil. So really most importations of produce, of plant material, of tractors and other equipment that might have been used in farming systems, one of the basic characteristics is you've got to be free of soil, or as free of soil as you possibly can, because that has the capacity of carrying nematodes. Obviously, in the early days, things would go by ship and take 3-4 months to arrive here. Nowadays, they can come by aeroplane, and so that is no longer the same impediment in terms of transport time. So the risk is greater now of bringing those pathogens in than it perhaps was 100 or so years ago. But even between regions becomes important. These things are not airborne. These things obviously move slowly through the soil system. So if you've got an infected paddock or an infected part of a paddock, try and avoid soil movement from there elsewhere. If you can keep it contained to one field or keep it contained to one farm or one region, you're actually going to minimise the damage. So again, that's all about movement of soil. Monitoring populations and understanding what's there and what's increasing allows you to at least make a risk assessment. If you know what levels are present, you can then make a determination on the likelihood of impact. There is a program particularly run out of South Australia where they test for a lot of the nematode pathogens in cereal systems. So some of the cereal cyst nematodes are really, really damaging. So before they go and sow their crop, they'll actually do soil tests. So they'll actually go into the field and they'll do a bunch of soil tests and they'll determine what the population level is prior to planting. And then they can make decisions. They can decide, do I plant or not in that field, or do I go somewhere else? If the numbers are through the roof, you're going to get a problem. You're not going to make any money. Why bother? Let's go somewhere else. And so you can determine your risk by now with some of these more molecular tests that can actually tell your soil population levels. Crop resistance is really, really valuable, really important if it's available. These things tend to be tough and they tend to be hard to be. A lot of that interaction is less direct. The more intimate the association between the pathogen and the host, the more biochemical interactions, the more capacity for resistance in a lot of ways. So these guys are a little bit cruder in the way they feed, a bit like insects, and so the level of resistance there is some there, but not necessarily all that wonderful. Where present, that can be quite useful. What's some good examples? There are nematode-resistant fruiting rootstocks, for instance. So a lot of fruit trees, or some fruit trees at least, they'll actually graft a nematode-resistant rootstock to the fruiting scion. So it'll produce the fruit you want, but you'll use this much more tougher rootstock that'll actually minimise the damage that the nematodes that might be present would have. Whereas if you put the fruiting wood as a root system, it's going to get severely damaged. Rotation can be really important, and it depends on the nematode. This is where taxonomy, I hope you've gathered I hate taxonomy, but it is important. Taxonomy becomes important because not only the type of disease they might cause, but also how you manage them. Some of these nematodes survive 1-2 seasons. Some survive 7, 8, 9 seasons in the soil. So knowing that will allow you to understand how well your rotation, or how quickly your rotation may work. So if you're pulling out maybe a vineyard that's had nematode issues, how long do you spell it before you replant with something new? It might be 1 year, it might be 3 years, depending on what the risk is or what the pathogen is there. Or if you're dealing with a lesion nematode of a particular crop, then you need to understand not only the rotation length between that particular crop, but also whether that particular nematode can infect some of the other species in the rotation. So for instance, if you're in a vegetable rotation and you've got a potato cyst nematode, that's going to survive in the soil for a really long time. So across a rotation, it won't solve the problem. If you're dealing with lesion nematodes, they are less long-lived, but they might infect potatoes, but they might also infect carrots, they might also infect something else in that rotation. So the choice of the rotation crop then becomes important. Not only the length, but the type of crop you use in the rotation to make sure you actually are getting a host break, not a crop break. Nematicides. They work relatively well. They can be right effective, but they are nasty. They are very toxic. They tend to be in the S7 classification, which is generally being thrown out by the authorities because they're too dangerous to use. So there's less and less of them all the time, and so those number of tools are really running out. And really they're not the most beneficial things to use anyway. They tend to be very nasty, very toxic. They'll kill nematodes. They'll kill most other things in the soil too. A lot of the fumigants as well. And they won't necessarily leave you with a terribly healthy soil afterwards. Biological control can be useful. As I said, you can actually get nematodes to eat nematodes. You can also get fungi that'll eat nematodes. How effective that is, there's not a huge number of commercial products that are really out there, so it's not that great, but it is an option. And suppressive crops. So this, I guess, feeds back into that discussion about rotation too. Picking some plants in the rotation, or in this instance a biofumigant that's not actually a crop, but it's actually something that actually produces essentially a mild soil fumigation as you're growing it and incorporating it, which can be toxic to the nematodes present. Right, so to finish, I think this is close to finish, I hope. What are some of the nasty ones? What are some of the ones we really are interested in? The root knot nematodes are major problems in a whole range. There's fruit crops, vegetable crops, pasture crops, field crops. There are a large number of different species, and they cause some major problems. This is kind of a fun one. This is associated with ryegrass staggers. Why is it fun? Not because it's fun on what it does to the sheep, but it's fun just because it involves almost every order of organism you can think of. So you've got a sheep, you've got an animal there, which is feeding on a plant, ryegrass, and it picks up a bacteria in the seed head, and the bacteria is producing a toxin, but it needs a virus, this bacteriophage associated with that bacteria to produce this toxin, and that's what feeds and causes the ryegrass staggers. But the bacteria is a soil-borne bacteria, and it's got to get to the seed head somehow. How does it get there? It gets carried up by a nematode. So you've got a bacteria infected with a virus being carried by a nematode into a plant, eaten by an animal, causing a problem. Kind of interesting how that ever evolved, that association. Anyway, lesion nematodes, big problem in the wheat belt, these lesion nematodes. Similarly, cyst nematodes in cereals are a huge problem. Potato cyst nematode is a very nasty one. We don't have here, thankfully. We've got it. It was in WA. They successfully eliminated it from WA by turning all those infected potato paddocks into houses. So shut down a whole bunch of people's businesses. It's currently in Victoria, and it's not. They're not doing that there, so it's still there. That's the major reason why spuds are not imported into Tasmania, is because they're likely to bring that guy with them. With cyst nematode, there is actually two of them. In Australia, we've only got the golden cyst nematode, or in Victoria. New Zealand's got both, and they are quite nasty. And that's what it can do, which is really cause major damage to your crop. And I was talking about all these cysts. I was hoping I had a photo because I'd forgotten I had one. So here's some pictures of cysts that are produced on the root. So this is actually a resting structure of the nematode. This is the golden one, obviously. This is the pale one, pallida, the white nematode, cyst nematode. And these get released by the infected plants and live in the soil for the extent of the next rotation, and then infect the plants subsequently. It's kind of fairly broadly distributed. What else in Tasmania? The bulb and stem nematode, Dilenticus destructor. is probably a bit of a problem. Dinolecus dipsicate is probably more of a problem in particularly onion crops and flower bulb crops. That can be cause big problems. These root knot nematodes and a whole range of veggies. Pretolenchus, the lesion nematodes, and again a whole range of crops. There's actually some great data for what it does in pyrethrum that no one even knew it was there, let alone causing any problems, until someone actually bothered to look, and then now they've been a bit more excited about it. And cyst nematodes, again, in a range of crops. Okay. So, there are a lot of nematodes, a good diversity of nematodes. Most of them are either beneficial or at least non-parasitic on things. About 10% are the ones that we're interested in this course, and they're the plant parasitic ones. All of them have a stylet, so that's the key thing to remember in terms of diagnosis. When you do your extractions and get your little jar full of nematodes from your soil, and you stare around the microscope and trying to work out, am I dealing with a plant parasitic nematode? You look at their faces and you actually try and find the presence of a stylet. If you have got a stylet there, you know at least you're dealing with something that is plant parasitic, because all of them do have that. They can be ecto, semi-endo or endo-parasitic. They can be sedentary or migratory, and they can be really hard to detect. Certainly from symptomology, it's usually not enough. You're going to have to go there and you're going to have to take samples. You're going to have to look in those samples for presence of nematodes. Okay, any questions?